Good morning, and as mentioned, my name is Ouso. And today I'm going to talk to us about using machine learning to model breast cancer patient survival outcomes as a way of discovering biomarkers that might be useful in precision breast cancer management. I'm based in the School of Computer Science of UCD under the program of the Center of Research Training in Genomics Data Science. Now, what are the challenges that we experience with modeling breast cancer outcomes? One is that most breast cancer modeling, outcome modeling approaches are faced with the challenge of covering a subset of the signaling pathways that are associated with breast cancer. And as you can imagine, there are a number of pathways that are associated with breast cancer, or any cancer for that matter. But then the reasons of things like dimensionality in genomics make it a bit difficult to deal with all this amount of data. The second thing is that due to those many signaling pathways or networks, there is a lot of heterogeneity in cancer, so that there's variations that differ from person to person and within a cancer in a person. And that makes it difficult to model outcomes based on genomics data. And so the key gap we seek to address here is that while most modeling predictors out there are associated with just picking a few of the genes associated with breast cancer or a few of the pathways associated with breast cancer, we are taking a global perspective of near all known breast cancer genes and pathways to incorporate it into a model to understand outcomes, survival outcomes in breast cancer patients. And so our objective is having that broader picture of breast cancer in a patient. And so how are we looking at this? Majorly, we are approaching this in two ways. We are using survival models that have been used for a long time in cause to effect models for survival. Another thing, we're incorporating explainability, which is an elusive thing in modeling biological systems. And so with that, we try to ensure that we can have some discoverable properties in the outcomes that we get from models. And so the Kaplan-Meier is a common one. And we use the Kaplan-Meier in helping us to dimensionality reduce in a manner that still maintains the features so that we know which features we're working with, unlike PCA or any dimensionality reductions that do away with the explainability in terms of you don't know actual features that you're dealing with. And then we also use the Cox proportional hazard model to understand, which is also an interpretive model to understand the effects of these features on overall survival. And finally, there is the aspect of the explainability that we are using the Shapley explainers to understand the contribution or the impact of the various features in the outcome of patients. And features for this case are genes. And so we begin with expression data, which are basically the fold changes in gene expression for various genes in a patient. And we collect that data from TCGA, is an open cancer data. So pick that from ETGA. And then we use the pathway databases like Reactome pathway to collate the different genes that are associated with different signaling networks for breast cancer. And when we bring that together, then we employ the KM, Kaplan-Meier, to get the expressions or gene expressions that are having a variability that is sufficient enough to help us discover something, some pattern from those expressions of those genes. And followed by that, then we are looking at certain pockets of gene expression that basically mimic the natural gene expression profile in humans, which is basically low expression. We have high expression, and there is also optimum expression. So with that, the Kaplan-Meier survival scan also help us to triage genes in these two categories of overly expressed genes and underexpressed genes, and some genes that show an interesting feature of both showing overexpression in a subset of patients, but also underexpression in a subset of patients. Then, what are some of the results that we come up with from when we've had these three triage categories of gene expressions? And we use this with the Cox proportional hazard model to model survival outcomes with these three particular subcategories of genes. And then after that, we then combine the outcomes from these three initial models into a single overall model by simply selecting for the genes that had the highest contribution to those individual categories of gene expression. And so with that, this is an explainer, sharply constructed explainer that try to rank the genes in the order of their impact to survival. So it ranks them from the gene having the highest impact on the outcome, which is overall survival in this case, and list them in the decreasing level of impact to the outcome. And so out of these four models, the very first one to your left is was a model trained on the genes that were showing overexpression only. And the second one is the genes that were showing underexpression. And then the third one are the genes that were showing both over and underexpression. And the final one is the model that picked the features that were high scoring or having high impact from the three previous models, and used that to then generate a fourth model that had those concordance indices, if you may, or we call them accuracies as a proxy. And then so we finally identify some 28 genes that are not causing, but are showing correlation to high or longer survival. And then in some of this, we get DK4 and Kremen1 as some of the genes in that list, which are known to associate to positively or in a helpful way impact the WNT pathway, which is associated with the proliferation in cancers. And so how is this important to us? Now, this is important because certain times when, for example, you're doing drug trials, you might be interested to trial within a cohort or a group of patients that show a particular profile in their expressions. This can be useful in identifying such a group of patients. And then there is the explainability that allows us to get some, to rank or get the impact of genes to the disease, which can then be interrogated further through experimentation in the lab or further evaluation to understand what their contribution is to the disease. Because I must mention clearly that the coefficients or the features that you get are not necessarily causative to the effect that you see. So that is some caution we have to have in mind. And then, of course, in doing that, we reduce the amount of time and the cost in trial processes or any experiments for that matter by narrowing down the number of potential genes that you need to evaluate to understand your disease state or your goal of interest. And of course, overall, if this is to work perfectly and we get a perfect model, then we are looking at the ability to tailor treatment for patients so that then, for example, we can tell that if a patient X comes and a patient X shows this profile for expression for the particular genes, then we can tell from the ranking for production state model, you tell that, okay, these genes are the ones that are dominating the disease of this state for this patient, and that these drugs that target these particular genes, how these particular genes function, and therefore you tailor the treatment to the patient. And with that, we are expecting a better outcome for that particular patient. There's challenges, they'll always be there. Some common ones which I may not have to mention is the issue of there is always almost never enough data. So that one, let's just put it aside. But I'd like to bring to our attention that beyond breast cancer, we can evaluate the possibilities with this in other cancers, because generally the dogmas of cancer are more or less the same. The hallmarks of cancer are the same. And so some of these findings may be transferable or maybe borrowed to other type of cancers. Next is complementarity in terms of integration of data. So there is the aspect of when we add expression data for this from RNA, we didn't have equivalent proteomics data to incorporate into the study. So that's a big challenge. And of course, for survival analysis, you need the longer the time, follow-up time, the better. And of course, having a standardized way of generating this data then allows us to compare with other data sets or incorporate other data sets to increase the amount of data that we have. And finally, the final challenge with this is that because biology is complex, sometimes there are confounding factors that are hidden within the system that need to be interrogated more deeply to understand really, especially when you want to now look at causality, to understand what, how these particular features that you have identified really contribute to the effect that we are seeing. And so what are some of the ways forward or to summarize is that by integrating all this, a lot of these signal networks, then we are accounting for a lot of the heterogeneity that exists in cancer, and therefore we have a more clearer or a better understanding, can lead us to a better understanding of what the cancer is about. And then, of course, we need to still evaluate this on real data, real-world data, and to validate those biomarkers that are proposed to be correlated to survival, to understand really what their impact is. And of course, this lays ground for very exciting times in AI, in precision medicine, and in improving healthcare as a whole when these things work. Otherwise, thank you very much. These are my associations, and I'd like to appreciate them for their contribution to this research. Thank you.
Thank you also for your presentation. We'll take 2 questions.
Thanks for a really interesting talk. I have 2 questions. So first question is a yes and no kind of thing. So, so what you did here is you ran a Kaplan-Meier or Cox proportional hazards through ordinary features, yes or no?
No.
Okay. All right, so you select a set of features using the Cox proportional hazards, or how do you select your features?
Select using the Kaplan-Meier.
You select using the Kaplan-Meier.
So all the 800 and something genes that are associated with the networks that you were looking at, we ran all those through Kaplan-Meier to select.
So that still brings my second question. That's what I thought. So the second question then, how do you control for false discoveries? And yeah, especially because you've got a lot of features there.
Yeah, there's a lot of features there, but then what's happening here is that we are not doing a classification problem. It's a regression problem. And so false discovery in the sense of positive or negative is not contributing here. Nonetheless, when you're talking about false discovery in the sense of a feature, you mean? Did I get you right?
Yeah, you essentially have multiple comparisons problem.
Yeah.
And with that, you have a probability of a false discovery, essentially.
What do you mean by false discovery? You mean like I'm including a feature that should not be included?
Correct.
Like the false positive, false negative.
Sorry?
The false positive and false negative. That's false.
I'm not sure that's the problem
We can
I'm
talk
solving here,
offline.
but
We
maybe
can talk offline.
I don't understand, or maybe, yeah.
Thank you very much for your talk. I'm Alina from Research Center Munich, Germany. I was wondering, so you chose SHAP for explainability. I was wondering whether you tried other explainability techniques, and if not, why did you choose SHAP?
Yes, good question. Thank you. We only tried SHARP for this, and didn't try other explainability options which are there, which is probably some next thing to look at. But SHARP, we chose SHARP because SHARP is. Do I remember the explanation for this? Sorry, I lost it.
Okay, thank you. Yeah, just wondering whether basically, like, if you try different explainability techniques and they obviously lead to the same result, that would be a really very good thing to see. Thank you.
Yeah, thank you.
Okay, thank you very much to our speaker. Let's give him another round of applause.